The production of thyroid hormone is complicated. A great number of steps have occurred before you end up with active thyroid hormone T3. Most of the T3 is made from inactive thyroid hormone T4. The thyroid gland itself produces primarily T4. T4 and T3 are synthesized on the inside of the thyroid follicles. The amino acid tyrosine and iodine are needed for the formation of the thyroid hormones T4 and T3. Iodine is ingested mainly via the salt in bread and other foods. Iodide, in combination with sodium, is actively pumped into the thyroid gland cell from the blood by the NIS, the sodium iodide symporter. After that, the iodide is brought to the inside of the thyroid follicle via an iodine channel, via pendrin and others. The thyroid gland cells produce thyroglobulin and also transport it to the inside of the thyroid follicle. Thyroglobulin contains tyrosines. The thyroid gland cells also synthesize enzymes, such as thyroid peroxidase, TPO, and the oxidases DuoX2 and DuoXA2, which are needed for the formation of T4 and T3. The DuoX2 and DuoXA2 enzymes convert oxygen, O2, into hydrogen peroxide, H2O2. Hydrogen peroxide is then used by TPO to oxidize iodide and to bind it to tyrosine. Tyrosine can bind to 2 iodine atoms. We call tyrosine with 2 iodine atoms DIT. T4 has 4 iodine atoms and is made when 2 DITs bind. Tyrosine can also bind with just 1 iodine atom. The result is an MIT. T3 has 3 iodine atoms and is made when 1 DIT binds to 1 MIT. The bond between 2 DITs, or between 1 DIT and 1 MIT, is also formed with the help of TPO and hydrogen peroxide. T4 and T3, bound to thyroglobulin, are stored inside the thyroid follicles. When the body needs thyroid hormone, bound T4 and T3 are taken up by the thyroid gland cells. In the cells, the hormones are unbound from the thyroglobulin, brought into the bloodstream, and are distributed throughout the body by the blood. Thyroglobulin contains many tyrosines. Not all tyrosines bind iodine. In addition, some DITs and MITs are formed which do not bind with one another to form T4 or T3. If thyroglobulin with these DITs and MITs is taken up by the thyroid gland cells, the enzyme dehalogenase, DHAL1, removes the iodine from these DITs and MITs. This iodine is reused for the production of thyroid hormone. The secretion of the thyroid hormones T4 and T3, which are stored in the thyroid follicles, is very precisely regulated. The hypothalamus in the brain produces TRH, thyrotropin-releasing hormone. This hormone ensures that the pituitary gland then produces TSH, thyroid-stimulating hormone. TSH travels to the thyroid gland via the blood and stimulates the thyroid gland to release T4 and T3, as well as to synthesize more T4 and T3. The thyroid hormone is then distributed via the bloodstream to all of the tissues in the body. When there is enough T4 and T3 present in the blood, the synthesis and secretion of TRH and TSH are inhibited. This results in a reduction in the amount of thyroid hormone being made and secreted. This way, the amount of T4 and T3 in the body is kept in balance. The thyroid gland primarily produces T4. T4 itself is not active. To become active, it must be transformed into T3, the active form of the hormone. This conversion takes place in all of the tissues in the body. The protein deiodinase removes an iodine atom from T4, creating T3. Thyroid hormone affects virtually all of the tissues in the body. The active form of thyroid hormone T3 increases the metabolism and with it the energy consumption. It increases the heart rate, the blood pressure and the rate of respiration. It stimulates longitudinal growth and the development of growth of the brain during gestation and in the first 3 years of life. And after that, thyroid hormone continues to stimulate the functioning of the brain.